University of Bordeaux, and he's also in France, and he's also very involved in France bioimaging. So please, Fabrice,
Okay,
there if you want.
so I will share. Oops,
I'm quite
my.
excited to see what you came up to with the 3D example. Fun.
Well, the thing is, I will give you a quick introduction to colocalization analysis, but I won't be talking about the softwares that you will be using, because basically you just have a look at the help section of your favorite software and you will find stuff about colocalization. The idea is to present some tools that already exist, some metrics and so on, and show you how they work and give you the time to imagine how you can apply it to your own research, to your own images. And thanks to the 3 datasets that I got and that I've reshaped a bit, thanks to Sylvie for asking for that kind of stuff, um, I will propose you to have a look at colocalization, but uh, on the side, using the usual tool that is quite nice in image processing, which is Fiji, and see how we can start building our own metrics, our own protocols about colocalization. I will switch from the presentation to a bit of either demo or follow along activities in during the 3 specific spots on this talk. Do not hesitate to stop me at any point if you missed something and you want me to give you a bit of a replay. But before we start, okay, the material is available. I hope that you've been able to download it. I guess that you got the link this morning. And if needed, the thing I can do is copy and paste the link to the chat so that you can do it. Please also open Fiji if you want to follow along. Otherwise, anyway, I will share my screen and you will be able to follow along.
Yeah, I couldn't share this in time, let's say, with the
Okay,
students. But,
sorry, yeah, I sent it this morning. So, okay, um, these are the things that I won't be talking about today, which are the controls. But they are super important, and I'm pretty sure that you already know about the controls you should do before starting any colocalization study. Of course, at the dice level, you must make sure that you don't have any bleed through or crosstalk. Otherwise, you would see the green in the red in the green, and it will be messy, and we will have plenty of colocalization, but not the specific one. Of course, you must make sure that you've got a co-registration of your channels so that when you're supposed to have colocalization, you do have colocalization, even though, well, when I'm talking about colocalization, I'm not always talking about having 2 labelings at the same precise position, but we'll see it later on. And of course, you must have knowledge about the resolution you're dealing with, because seen from the moon, all the countries are only one when you look at Earth. So, of course, if you're looking at colocalization from the distance, everything will colocalize. And colocalization is always stated relative to a certain resolution. If you increase your ability to resolve structures, of course, you have less and less chances to have colocalization. Colocalization is a nonsense because physically you can't have 2 proteins at the same location. So you must define the perimeter at which you will estimate that the 2 proteins are close by. That close by study is It's not as sexy as colocalization when you try to name it. And the second thing you've got to do before starting colocalization study is know precisely what you want to do. Do you want to have a look at two proteins that are not precisely at the same location, but maybe within a similar environment? For instance, if you've got a cell that is expressing a marker in the cytosol and another one in the nucleus, some people will define this as colocalization. They are not colocalized. Well, they are not precisely on the same spot, your two proteins of interest, but they are within a certain defined environment, which is a cell, which might be anatomical structure or whatever. And in that case, well, I'm more talking about co-expression. If I go down from the cellular level to the subcellular level, I can talk about co-occurrence. The two proteins of interest might not be exactly on the same spot, but they are in a smaller region, that is subcellular level. In that case, I will look for co-occurrence of the two proteins of interest. The two proteins of interest might be on the same subcellular structures with a link in terms of intensity, in terms of abundance. And in that case, I will look for a link between the intensities and I will look for correlation of intensity. And sometimes the two proteins of interest might just have a pattern that depends one on the other. And I will rather talk about co-distribution. So you see, in fact, colocalization is not a proper term because it all depends on what you're looking for. You're looking for a relationship between two proteins of interest relative to a reference system that might be the cells, that might be within the cell, that might be on a specific structure within the cell. And so please, before stating I want to do colocalization, especially if you're interacting with imaging facilities, just try to describe your problem and then we will try to find a proper metric in order to define colocalization. And in terms of metrics, I artificially divide the type of metrics into two big families, the indicators and the quantifiers. Indicators is just a numerical value that will evaluate the degree of coincidence of colocalization, well, whatever you name it, relative to a scale. You might have an indicator that goes from 0 to 1 and have a value of 0.9. It doesn't mean that this is 90% of colocalization. This is not related to a physical thing, a physical phenomenon. It's just a way to evaluate over a scale where you've got full colocalization on one side, no or even exclusion on the other side. And it might be hard if you don't have a second experimental condition to draw a direct conclusion. So, because when you're using a scale, you can compare two values, but if you only have one value and a scale, unless you're on extreme values, it might be hard to conclude. But we've got strategies, we've got tricks, even if you've got only one single experimental condition, to try to make sense out of a single value. And we will see it at the end of the talk. A quantifier is more something that is linked to a physical phenomenon. For instance, let's think about the overlap of the degree of overlap of two signals, the area that is involved in the colocalization phenomenon. In that case, it's easier, even though you might have a scale, it's easier to draw a direct conclusion about the number. Is it okay for everyone? This is a bit this is a bit trying to give a frame, which is one possible frame, one way to interpret colocalization and all the numbers that we can extract from the data. This is one view that I'm proposing right here. So when dealing with colocalization, we can work on intensities. We can work on objects. So first, I will show you what you can do with intensity-based approaches. And here is a graph and some math. Don't worry about the math. Here is a cytofluorogram, and this is a first way to work with intensities. Basically, each single dot that you see here has two coordinates, x and y, which are the intensity for this individual pixel on the image, intensity on channel 1, and on channel 2. It gives you one dot on the graph. If you take twice the same image, of course, the intensity is the same along axis channel 1 and channel 2, and you get a nice line. You've got the perfect case, perfect colocalization. Unfortunately, this is not what we are expecting to do when dealing with our own images and sample. If you've got, each time you see a certain quantity of intensity in channel 1, you've got, let's say, twice this intensity in channel 2, you will end up with something that is close to a line. And this, the relationship that you've got between the intensities of the two channels, is something that you can characterize using a coefficient that is called the Pearson coefficient. Be careful because, well, sorry. This is an indicator. This is what I wanted to say. This is an indicator because the correlation is expecting to be within a range of -1 to 1. So -1 is exclusion. You would have a cloud close to one axis because where all the channel 2 pixels do not carry any information when looking in the channel 1. And you would have a second cloud here because pixels from channel 1 would not carry any information on the channel 2. No correlation would give you a cloud close to the axis because there is no dependency between the intensities of both channels. And 1 would be the perfect colocalization here. You've got a perfect linear relationship between channel 1 and channel 2. I find it super nice when you've got several samples to compare. Once more, I've got an indicator, I've got a scale, and I can compare the two images. But what if I've got 0.75? Well, it's quite close from full colocalization, but not far from the middle range. So how do I conclude from that? We will see later on if I've got only one experimental situation, I will be able to generate additional information, additional control situations directly from the image. Be careful with Pearson coefficient. You see the scales start at -1 up to 1. 0.75 doesn't mean that you've got 75% of colocalization. It's just a value that say that there is quite a strong relationship, linear relationship between the intensities of both channels. Moving on to another intensity-based quantifier this time. We've got the well-known Mendel's coefficient. It requires first the user to identify objects on both channels. You would identify the red, the green objects. Then you would sum the intensity of all objects within the red channel. This gives you this. Then you would look at the intersection where both colors, both objects overlap, and identify, calculate the total intensities within the red channel, within the overlapping area. And by dividing this part of the signal over the total signal within one channel, you end up with one coefficient. You would do exactly the same for the second channel and end up with two coefficients. So this is a quantifier because it allows you to quantify the percentage of your total signal that is involved in the colocalization process. Unfortunately, it relies heavily on the way you will isolate, you will segment your image. And most of the time, people are using threshold. Threshold is something that is based on the intensity, and you're trying to quantify something based on the intensity as well. So be careful about how you would impair your measurements based on the way you're processing the image and doing your segmentation. Is it okay for everyone so far? Yes.
You guys can have questions if you want. Anything?
Is it something that you already know, I guess?
But we haven't spoken at all about it, so I think that, but there are several people who, as you know, who use, and it's always a little bit blurry what to use.
Yeah. Okay, if I've got no question, I will move on to another way to work on intensities when I've got discrete signal. You see, I've got, so I'm not sure that you can see my the cursor, the arrow. Can you see it?
Yes, we can see your pointer.
Yeah. It's
Perfect.
really dark, but we can see it.
Okay. So you see, you've got some comma-like structures on the two images. They are partly overlapping. They are small relative to resolution. So trying to extract the part of the signal that is associated to the small structures, it would be quite difficult and prompt to errors. So my colleague, she came up with a method, a small, just a nice method where, in fact, you would draw a line across the two dots, the green, red one. You would plot the intensity profile in the red and the green channels and look for the overlap between the signal. And if this overlap goes beyond the optical resolution, it means that I'm not able to distinguish to distinguish apart the two structures. They do overlap, so this can be scored as one event of colocalization.
So when you see beyond, you're saying smaller.
Above, far above.
So
If you've got
if the distance is larger than
Yeah.
the point sprite function, then we say yes, it's colocalization.
Yeah. in fact, the problem is what we should draw as a conclusion is, considering the resolution, I'm not able to distinguish the two objects. Therefore, as a biologist, I conclude that there is colocalization. But in fact, the true conclusion of a colocalization test is, according to the current resolution, I'm not able to distinguish. I can't exclude that there is colocalization. See what I mean? The conclusion is, as biologists, we have to conclude something, but in fact, maybe the real conclusion is I don't have enough resolution to be able to distinguish the two objects. Okay. Let's experiment together. So I will take the images from Uria. I'm not sure that I got right all the images that you sent me and that I've been spotting the right area within your sample to do colocalization. And so if I consider, so this is a full image, if I consider a zoom on the image, I've provided only the zooms so that it's easier to handle on our computers. So you see that I've got a discrete green signal over this fiber, this wavy structure that is spanning in 3D. If I want to explore the colocalization between the patchy structures in green along with this wavy continuous structure in red, how would you proceed?
Any suggestion? I think it's a nerve, by the way.
Yes, it's nerve.
So how would you do that, Aurea?
Sure, I need like to have the whole structure, so I need to do like maximum intensity projection to see the whole nerves.
Mm-hmm. So maximum intensity projection, and then what would you do?
Yeah, and then see if I have colocalization or overlapping of my two proteins.
Okay, overlapping between the two proteins.
Yeah.
You see, the drawback with maximum intensity projection is, let's imagine that, and unfortunately, you've got to look at me there, because I'm miming the stuff. If you've got the nerve there and you've got the dot below, when doing maximum intensity projection, one will fall on top of the other. So you would conclude that there is colocalization, but there is no colocalization. So I'm proposing a slightly different approach. And this is the kind of thing that we will be able to do together. So how will we proceed? First, we will generate an appropriate view. And second, we will use intensity profile to investigate correlation. And here is how to do it. So you've got a slide with everything that is explained and the functions that I'm about to use. Okay, let's go.
So we should open the presentation or something like this to get the explanation?
Yes. So if you want to follow along, the thing you can do is open dataset number 1. So it's a crop, and the
if you go
colors
to
are
the lecture colocalization over there, I think you have the presentation, and then you can see the slide. You can download the PDF of the lecture, then you can see it.
Yeah. So in here, the thing I will do is I will go use right-click on the linear tool, go, let's say, to segmented line.
Can you, sorry. Oh, yeah, now I see, yeah.
Just to prepare. But if I move along the z-axis, I see that everything is not in the plane. Therefore, I will do a maximum intensity projection. Not because I will work on the maximum intensity projection, but because it will allow me to follow the nerve, the track of the nerve. To do that, I go to Image, Stack, Z Project. I will use all the slices to a maximum intensity projection, meaning that I will create a new 2D image where I retain over all the 35 slices for each pixel only the highest intensity pixels. Now I see the path. I could play with brightness and contrast, but I won't do that. And now using the segmented line, I will follow the track and double click at the end. The idea will be to transfer the region of interest that I've got here onto the stack. To do that, I use Edit, Selection, Restore Selection, or the shortcut is Shift E. So same location, but on the original image.
I just want to mention that if you do not see the segmented line, you can click on the small or a red triangle next to the straight line, and then you will get segmented line.
Right click on the button and you will get access to all buttons. The thing I will do is I will actually double click on the tool, segmented line tool. This allows me to define a certain width for the region of interest. And you will see it will be quite useful. So we know the path. We know that we should collect pixels along this path, but we will have to do it along the z-axis. Just imagine that I've got a big knife, and this is a track along which I will go with the big knife to make a cut through the stack. I will do it using the image, stacks, and reslice function. Don't worry, just click on OK. And oops, here, what you see is a cut along this line, along the z-axis. So in X, you've got the line, and in Y, you've got the z-axis. Okay? I just have been popping the brightness and contrast box to show you what I've got. So now the thing I can do is redo exactly the same, draw the path along which I will do the analysis. Same principle, use segmented line and draw the line. There are tools that allow you to ease this segmentation process, but it's also important to show you how you can do it manually. Currently, I can navigate from channel 1 to channel 2. So channel 1 is red. And now I will go to Analyze and Plot Profile. This is the intensities collected along the track, averaged on the width of the track for the red channel. If you want it to be more easy to remember, within the graph, you can go to More, Content Style, color, let's say red, line width, I go for 3. Is it okay for everyone? The one that follow along, the one that
I have a question.
do
I
not
have a
follow
question.
along, but just to
When
Yeah.
you were doing the profile that it was a bit expanded there, this reslice stuff, I just get one line, so I cannot zoom in. It's just one line. So I was wondering what type of output spacing in micron start at the top. What type of options you had there?
So I guess that you've been trying the reslice directly on the projection.
Right. Yes, you're correct.
So first you have to activate the original stack.
Yeah.
Then edit selection, restore selection, or Shift E.
Right. Yes, that's it.
And then reslice.
Yeah.
So I've been doing the profile in red. I will simply move the slider here to activate the green channel. And it will do exactly the same graph when I'm calling the process, analyze dot profile. It will do exactly the same, collecting the intensities along the path on the active channel. Here it is, green. If I want it to be a bit clearer, then More, Content Style, color green, and line width, I put it 3 so that it's easier to see on the screen. So if I see that I've got peaks, are they matching peaks between the two? I would need to overlay both graphs. So of course, here on list, I've got all values. I can export it to whichever software you like and do the overlay. But we can also, here, if I go, it doesn't matter if you do it on the red or green graph, but if you go to data, down on the list, you've got add from plot. So I use add from plot. And then select a graph from another plot. And here it is. I've got an overlay of both. Of course, the scale is not the same. So I should have addressed this problem before. But I can already see that there seems to be some correlation, some stuff going on. So now, how do I analyze precisely this kind of stuff? Well, first, I always have the correlation coefficient. I can always go back to the Pearson coefficient, for instance. But here, I will be, it will be more precise because it will be dedicated to the single structure that I'm currently analyzing. Instead of having a tiny bit of colocalization that is within a huge image of non-colocalization, I've been focusing on the region of interest, and now I'm able maybe to unmask a discrete colocalization. So this is one way to go. Another way is I could be quite inventive about how I express colocalization. I could count, let's say, the matching peaks and define that this is my metric. Of course, if I want to compare two nerves, I must make sure that I'm comparing the same length of nerve. So maybe having a number of events divided by a length of structure would be another metric. The thing I'm trying to show you here is that we already have metrics that are well generalized, but are they adapted to all situations? The answer is no. So try to see what you can extract, what is relevant, and maybe invent a new way to do your own colocalization. Is it okay for everyone? I'm sorry, I'm just rushing a bit because the time is tight. But I've been putting all the steps so that you can redo it if needed. Okay? Let's see. Yeah, I will re-share that one. Okay, object-based methods. So object-based methods, there is a modified Mendel's coefficient method that exists. We've been isolating objects in the red and in the green channel. We can evaluate the what is going on in the intersection part of the object versus the whole object. And we've been evaluating it previously in terms of intensities, how much intensity, what percentage of the intensity is involved in the colocalization. But you could think about doing exactly the same about the area or about the volume. And you would end up with two ratios, one for the surface of or the volume of red objects that are involved in colocalization, and same for the green part. This is called modified Mendel's coefficient. This is one way to do it. But what if I've got small, tiny objects? Let's say objects that are close to the optical resolution. If you've been doing the sampling the right way, one tiny little object will appear on 3 pixels per 3 pixels. So in terms of overlap, you would have 1 pixel out of 9, or 2 pixels out of 9, or 3 pixels out of 9, etc., etc. So you will have a discrete estimate of the colocalization. In that case, overlap is a total nonsense. So you have to end up with something different. And one way, and especially if you've got a difference of size between one channel and the other, the thing you can do is for the tiny little object, which is not the case on the illustration, but don't mind it, you could think about the tiny little objects in red and summarize them as their center. And in that case, you would build a different metric, which is the center of objects in red falling onto objects in green. And in that case, you would count the percentage of centers from red falling onto objects in green. Be careful, because when using this and having some situations where you've got a ring around a structure, the center of the ring, in that case, would fall onto the object in green, which is a problem if you want to estimate the colocalization, but which is helpful if you've got a wholly signal to be compared to something that is filled. You see, you don't have any overlap, but center of red is falling onto green, meaning that red is around green. You see that when thinking about the metrics that you're building, you have to use maybe your metric about colocalization and additional external metrics so that you draw a conclusion. No overlap, but coincident between center and object, meaning that one signal is around the other. You could think about summarizing everyone as their centers, and this is quite useful, especially if you've got small, tiny objects. And in that case, you end up with dots, and of course the dots won't overlap. It will be harder for the dots to overlap. Then you should find something else to conclude about colocalization, and this something else is your resolution. If the two centers are distant from a distance that is more than the resolution, then you can resolve them, then they are not colocalized. If the two centers are apart from a distance that is smaller than the resolution, you can count this as a colocalization event. And here you've got another indicator, quantifier, sorry. Okay, so I won't go, I'm sorry, Ellie, I won't go for the demo on this example. Anyway, I've written the procedure so that you can try it at home. So here, it was quite hard for me to interpret the data. And I assume that what you were looking at is small dotty-like green structures and big red structure. Am I right, or?
Yes, that's right.
Okay. So in that case, what would you do?
Shall we segment the big one?
Yes, segment the big one. And what about the small ones?
Ellie, do you know?
Well, about the small ones, so about segmentation, we will segment. We'll extract some information about the two channels, but it will be segmentation that is differential from one channel to the other. We would segment the objects, and then on the green channel, we will identify the centers. Or more precisely, the small dotty-like structure like that, they can be detected with a nice tool in ImageJ, which is find maxima. The small dotty thingy is like a local maximum over a dark background. And it's quite easy to isolate this as long as the background is homogeneous. So in few steps, and you will be able to do it, hopefully, because you've got all the functions that are described here. I would separate the channels. On that one, I would apply a threshold, even if it's not the best method for segmentation. We can always discuss about that. I will end up with a mask where I've got black pixels in the background, white pixels on the objects. On channel 2, I will use, this function that is local maxima detection, so process find maxima, and use an output that is single points. It will give me little tiny points where I've got local maxima. And then how do I evaluate the centers falling onto the objects? Well, it's quite simple. I will use process image calculator and use a logical AND operation. I will create a new image where pixels are turned on only if they are turned on the first image and on the second one. This image is the intersection between this mask and the dots detection. I know that out of the 6 dots I've got here, only one is falling onto the red object. This can be my metric, counting the number of remaining points over the total number of points. Maybe you're interested in a per cell count. As long as you're able to identify the cells, then you can delineate it and count the number of objects in the end mask within the cell. Is it okay for everyone? Yes?
Yes.
Okay. Now, the really last part, and I'm running out of time.
Yes, but it's okay because we I think we're quite done with the quizzes that we're supposed to do afterwards. So I think you can have another 45.
Okay, perfect. Comparing. So I told you when you want to do colocalization, it's good to have a control situation and your experimental condition so that you know the range, you know where on the scale you are located. But it's not always possible. And sometimes you want to have an absolute, let's say, absolute diagnostic about colocalization. So how do I do when I only have one data set? I will have to generate a new synthetic data set where the content is exactly the same, but it's shifted around. So you've got to break the colocalization if it exists so that you've got the zero of your experiment. And one way to do it is simply by taking your 2 images, making whatever measure you're happy to do, and then you take image A as it was, but image B, you just tilt it 90 degrees. This is the old way to do it. This works only if you've got a tiny little number of objects. If you've got a crowded surface, then it will be harder to use this as a control. Another way is to randomize objects or to randomize pixels, not simply doing the rotation, but really picking, for instance, the green objects that I've got here, isolating them and putting them back on the image at a different location. This way, you've got same content, but you will be able to estimate the colocalization that you end up with that is due to randomness. And if when you're comparing your original images to this random distribution, your original data fits within this distribution, it means that it might be colocalization, but it might just be a random process that ends up giving you colocalization. If not, if it's far away from this distribution, then well, happy you. Okay. And I go with the images, those nice images to illustrate this. And once more, the thing I will do is show you, and I wanted to illustrate with this specific example, the randomization. What I've been preparing for you, and especially for you, is a small image macros that does the job. First, what I would do is segment the cell. Try to find the outlines of the cell. And in the macro, it's not the best way to isolate the cell and so on. Once I get the outlines of the cell, which is my perimeter on which I will isolate the objects and randomize the object, then I isolate the objects. And as a second step, I take my objects and I throw them within the cell randomly. Okay. To do that, okay, just to show you that it's going quite fast. To do that, I open the dataset. I will split the channels, image, colors, split channels. And I will exemplify only with one image. And just to make sure that everyone is able to see, I'm switching the color table. I'm opening the IGM image macro file. Okay, and I press the run button. Cell detection is done based on the threshold. It's not ideal, but it's there. I will then use the analyze particle function in order to delineate the cell, and I will only keep the largest object that analyze particle will have found. Within the cell territory, I will detect the individual objects. Once more, I'm using a threshold in order to segment the cell, and the minimum and maximum object size just to filter out small stuffs that are going around. When I will press OK, it will do the cell segmentation, the object detections, and the randomization. Okay, here it's copying, pasting the objects at random position on the output image. It's done. So these are the detections. You see the outline of the cell, so it's not perfect, but... And these are the objects that were detected. And basically everything outside is black. And from this black canvas that I copied here, I'm just copying the content of this region of interest and putting it here, for instance. That one, but you see the principle. The only thing that this macro is not handling is the overlap between objects, because by putting back the objects within the cellular space, two objects might end up overlapping, which is not good. But well, I only spent 1 hour and a half on that one, so sorry about that. And once you have this new image, then you would compare, for instance, channel 3 with original versus channel 3 and multiple randomized images. With which metrics? Well, have fun. It all depends on what you're expecting. Maybe in that case, I would summarize the object by a center and work on center-to-center distance. Okay. Then, oops. Then, sorry. And it's time to conclude, but it seemed that
I'm sorry, can I ask something?
go ahead, and in the meantime, I will try to find back my window.
If I do the randomize, is it possibly for make it as a control for colocalization or?
This is totally a control for colocalization.
Okay, thank you.
That's the idea, is actually to create
Yeah.
a control that is random.
Yeah.
But I guess, for example, in this scenario you had here, you would also need to consider, for example, because here it was randomized to the entire cell, but the first one seemed to be only in the cytoplasm and not in the nucleus. And then the control would be much different because it's not randomized in the same cell compartment.
For sure. This is just to show you the principle. But then in that case, if you only have the signal in the cytosol and it's excluded from the nucleus, then you should have a labeling of the nucleus so that you can exclude it when doing the placement of your objects, randomized objects. So yeah, I'm happy because you see, this is what I wanted to do, which is make you think about how to do it. Well, and this is the last slide. I'm sorry because my.
Do you want me to share it? Because I have it.
Well,
Yeah.
please go ahead.
Can share it.
Yeah, my web browser crashed.
So I will share that.
Can I ask a quick question about this? So for this macro is one that you programmed yourself. Were there any specific functions that we can, for example, find on how to randomize this placement, or any good resources on how to do these types of macros on our own?
Yes. So to do the macros, if you want to do macros, just have a look at the NIS YouTube channel. This is, if you're interested in ImageJ and additional stuff, just go ahead and have a look at the NIS YouTube channel. If you're interested in image processing in general, automatization, Python, and so on, have a look at what Robert Haase, Haase, has been doing. He's been working quite a lot on providing material for this. So yeah, Unfortunately, Sylvie, it's a bit of a spoiler because previous slide
sorry.
was ta-da, the magic recipe that always works.
All right. So yes, that's
Okay,
true.
it does not exist. So please think about your own study case. Build the metric that best describes your situation. Validate it with simulation with additional experimental condition that you can control, and then share it. So I've been showing you things that already existed, things that are widely used, but this doesn't mean that it's adapted to your personal situation. And so if you've got ideas, if you can explore and be the next one to publish a colocalization metric.
So I have a question. In which situation would you then use the Manders and or improved Manders and the Pearson coefficients instead of doing your own analysis?
If I see that it is obvious there is a linear relationship between intensities, I will go for Pearson. And also, if I have to restrict to a certain area, I may restrict the analysis to a certain area. If I see that I've got big objects or I've got populations of cells within a tissue and I want to measure an overlap, then I would go for Manders. If I want something quick on an obvious case, of course I will go for things that already exist. But you see the little trick with the nerves, with the peaks and so on. I could have counted the number of matching peaks, for instance, and this is a new metric. And I'm pretty sure that Manders, it would be just crap in this situation. Pearson coefficient, maybe, but in that case, those metrics are not adapted. So now I've got to sit down, look at the images, see what I call colocalization, try to come up with a metric, validate it, make sure that it's working, and then say, okay, this is what I consider to be colocalization. If your case is obvious, go for the existing stuff, but don't get stuck with the old existing stuff.
That's a nice end message. So do you guys have more questions?
Thank you. Thank you, Fabrice. Thank
Welcome.
you for building the pipeline for me. I'm the one with very tiny dots and 4 channels. And I always, because my colleague always suggests me to do the Manders colocalization, but for me, because I have many channels and I will do the heteroprotein imaging. So for Manders, I think it's very subjective to select the threshold for me. And I also have different background for the dots. So I'm wondering if I can measure the intensity. Like if I segment one channel that I would like to measure and project it to other channels to measure other channels' intensity and to contribute to a possibility that for colocalization. But I didn't find the proper literature for that.
And you've got a really small compartment. You've got small vesicles that are quite close to the resolution. So unless you go to super resolution, I would say that measuring overlap, or at least measuring something that depends on the overlap, including the first definition of Manders coefficient, where you see the percentage of intensity that is involved in colocalization, might not be the best way to go. So I would definitely have a look at local maxima, because the local maximum detection depends on how big the peak is above the surrounding. Meaning that even if you've got a non-uniform background, it will be able to find the peaks.
Yes.
Once you've got this, you've got the coordinates of the detections, and you measure the distance between channel 1 and channel 2 and define how many couple of structures you've got that are of a distance below the optical resolution. And these are your events of colocalization.
Okay, and why it is better than measuring the intensity? Is it because the background stuff?
Because the problem is, if you've got an object that is close to their resolution,
Mhm.
when using the Shannon-Nyquist theory, you will end up with 3 pixels per 3 pixels.
Oh,
So
yeah.
only 9 pixels. So it's quite a low number to define precisely an average intensity or a total intensity. If you add on top of that a bit of noise, then you don't have enough samples, I mean enough pixels, to precisely define a global intensity for your object.
Okay.
So unless you've got the brightest possible and the cleanest possible labeling, it might be hard.
We go back to sample preparation. I think we have to stop this interesting discussion. So thank you very much, Fabrice.
Thank you. And sorry for taking more time.
Okay, I put you at the end of the day for some reason. So have a great day.